This video will instruct you on how to prepare the plates for saccharin or a control conjugate. Please refer to the PDF file on the flash drive included with the kit for complete protocol instructions. First, remove the vial labeled saccharin or control conjugate for whichever plate you're preparing. Then, pour roughly 10 to 15 mL of cell media into your well reservoir. Next, label 8 microcentrifuge tubes 1 through 8. Next, take a pipette and transfer 135 microliters of cell media into tubes 2 through 8. Next, vortex your sample of saccharin or control conjugate and transfer the calculated volume into tube number 1 and vortex. For saccharin, transfer 45 microliters. See protocol instructions to calculate volumes for control. For the saccharin plate, transfer 105 microliters of cell media into tube labeled 1. For a control conjugate plate, please see the protocol PDF for transfer instructions. Then, perform serial dilutions by pipetting 15 microliters from tube number 1 into tube number 2. Pipette up and down several times to mix. Vortex the tubes briefly. Repeat by pipetting 15 microliters from tube number 2 into tube number 3. Continue to repeat these steps through tube number 8. Once you've finished making your dilutions, you are now ready to begin adding the material to your plates. Begin by placing the well template underneath your plate. The first step is to add to the control columns. You will add 10 microliters of cell media only to each of the 6 wells, B through G, of each of the control columns 2 and 11. This means the first and last wells in the columns will not receive media. Then, starting with tube number 8, vortex briefly and transfer 10 microliters into each of the 6 wells to the column labeled tube number 8 according to the template. Repeat this step for each of the dilutions. Tube number 7 into column labeled tube number 7, tube number 6 into column labeled tube 6, and so on until tube number 1 into the column labeled number 1. Once you complete adding the material to the plate, place it back under the appropriate growth conditions for your cells for 72 hours.